Enzymes were discovered in 19th century when Buchner observed that the conversion of sugar to ethanol carried out by yeast during wine fermentation still occur even when yeast are dead. For chemists, the study of kinetics of enzymes was attractive because they observed that the velocity of the reaction has a nonlinear dependence of the quantity of substrate. They know that the dependence could be adjusted to a hyperbola equation, but there was no justification for do this. The model that explains this velocity behavior was proposed by Michaelis and Menten. They proposed the existence of a complex formed by the enzyme and the substrate, and this is the famous equation that describes the model. But how to get the equation from the diagram? There are two ways to derive the velocity equation. The first method is known as fast equilibrium. To understand equilibrium, we can consider two water reservoirs connected and separated by a hatch. At initial time, all water is in reservoir A. When the hatch is open, the water flows to reservoir B until both reservoirs have the same amount of water. This is the equilibrium state. In the enzyme diagram, we can consider E, ES, and P as three connected reservoirs, but the assumption here is that the flow of water from ES to P is limited by the thickness of the connection. In chemical kinetics, it means that the velocity of E to ES is much more bigger than the velocity from ES to P. When the hatches are open or the reaction starts, the reservoirs E and ES reach a condition called quasi-equilibrium. In the velocity equation, that is, the differential equation that explains how the product is formed in time, the term ES can be substituted from the value of the equilibrium equation. Doing some algebra and normalizing for total enzyme, the equation is derived. The second way to get the equation is by steady-state assumption. In this case, the state is reached when the communication between the reservoirs can support the same amount of water. During some time before the equilibrium, the amount of water in compartment ES doesn't change. It means that the rate at which it is formed is equal to the rate it is consumed, and the mathematical expression for this is equals to 0. Again, the value of ES is obtained from this equation and the Michaelis-Menten equation is obtained.